Welcome to the UC San Diego Shiley-Marcos Alzheimer's Disease Research Center Brain Blast series. My name is James Brewer, and I'm the director of the UC San Diego Department of Neurosciences and the Shiley-Marcos Alzheimer's Disease Research Center. I'm a professor of neurology, a clinical neurologist, and a brain imaging researcher. I'm pleased to present to you our virtual Brain Blast series. Each of the talks included in the series have been created by Shiley-Marcos ADRC colleagues with expertise in brain aging research that focuses on prevention, detection, diagnosis, and treatment of Alzheimer's disease and related dementias. Each of the experts featured in the series studies a unique aspect of brain aging and has developed a brief overview of their current research to highlight the state-of-the-art work that's going on here at UCSD and our center. We hope you'll take advantage of this free resource to learn more about the advances in brain aging, the importance of research, and the multitude of opportunities to get involved and participate in research studies.
Hello, everyone. Welcome to the next edition of the Brain Blast series. My name is Ken Diffenderfer. I'm the director of the Stem Cell Core at the Salk Institute, and we work very, very closely with the Shiley-Marcos Alzheimer's Disease Research Center as lead scientists for the Induced Pluripotent Stem Cell Core. Today, I wanted to share with you all some specifics about the incredibly novel patient-based cell models of Alzheimer's disease that we are able to generate and use for research purposes to unlock the mechanisms of AD and also provide an important platform to possibly identify better therapeutic solutions for the disease as well. So let's get into it. At the center of the core, our mission is to provide Alzheimer's Disease Research Center researchers access to state-of-the-art human cell-based models from the longitudinal cohort that the ADRC works with. We do this primarily through accessing cells through a dermal punch biopsy. Longitudinal participants in the study can consent to this process, and a very small piece of skin is removed from the back of the forearm in a very minimally invasive procedure that leaves minimal to no scarring. And from that little chunk of skin, which is about 3 millimeters in diameter, we can treat that biopsy material overnight with enzymes and start to break apart that tissue and ultimately extract the cells that comprise the skin from that. And these cells that we generate are called dermal fibroblasts. Dermal for skin, and fibroblasts are a unique cell that we see in many parts of the body, especially linings and layers of tissue. And this dermal fibroblast is the starting point for all of the disease modeling that we do inside the IPS core, the Induced Pluripotent Stem Cell Core, for the ADRC. The two primary models that we intervene with in this modeling endeavor are induced pluripotent stem cells, which I will go over in detail momentarily, and also a new modeling approach called direct conversion technologies. Both of these approaches start with the fibroblasts that we just discussed. So let's move on to induced pluripotent stem cells. First, I will provide some background here so we can all appreciate this technology and the impacts of it. To go back a little bit, very, very early in human development, there is a unique cell, a unique stem cell that has the ability to generate all the cell types in the human body. And we can imagine this cell as a ball at the top of a large and undulating hill. And we can imagine the process of human development as that ball being nudged from its perch at the top of the hill, rolling down the hill, and hitting key points in the hill, which determines the direction of the ball. Ultimately, that ball is going to end up at discrete locations at the end of the hill, and we can imagine these as the distinct cells and cell types that make up the complexity of the human body. This process in developmental biology is called differentiation. And for the longest time, this process of development, this process of differentiation, was thought to be a one-way street. So essentially, as the ball rolled down the hill, there was no way that it was going to roll back up, right? That just could not happen. It would not happen. In 2006, though, a Japanese scientist by the name of Shinya Yamanaka kind of turned the scientific world on its head and showed that through a process of reprogramming, that these permanently differentiated cells, a permanently differentiated cell, a permanently developed cell that has made all of its life choices could be reverted or reprogrammed back to the very top of the hill. Dr. Yamanaka named these cells induced pluripotent stem cells and really unlocked a brand new arena of research that could provide a powerful platform to model human disease. And that is all captured here in this somewhat complicated schematic, but I'll go through it at a very high level here, just so we can all be on the same page and appreciate this process. And so again, it all starts with the patient. It all starts with the unique individual with a unique genetic background. We can generate fibroblasts from those individuals using the punch biopsies that we talked about earlier. And then from there, we can introduce 4 key genes into those fibroblasts. And those 4 key genes kind of reset the cell and make it think it is something very different. And they push that permanently differentiated fibroblast all the way back to the top of the hill. And at that point, we have something that we call an induced pluripotent stem cell. And the real power of these induced pluripotent stem cells is that we have, recreated that stem cell that exists very early in human development. And now using other techniques and tools, we can control how that cell now differentiates. And ultimately can use this as a very special and unique platform to generate specific cells like those that exist in the human brain and are involved in the pathology of Alzheimer's disease. So neurons, astrocytes, and microglia are 3 of the key cell types we are interested in when we talk about Alzheimer's disease. And so if we think about this process in the context of AD, in the context of Alzheimer's disease, we can have participants that have been diagnosed with Alzheimer's disease. They can have consented for a biopsy. That biopsy can be used to generate fibroblasts. Those fibroblasts can be reprogrammed into induced pluripotent stem cells, and ultimately those induced pluripotent stem cells can be differentiated into the cell types that are involved in the in Alzheimer's disease, into neurons, astrocytes, and microglia. And the center of this technology is throughout this entire process, we have captured genetic information that could be impactful for the progression of this disease, and are capturing that in a cell-based model and in a dish, and can do a lot of fun and interesting research to interrogate what's happening at the molecular and cellular level using these cells. Another really cool element of induced pluripotent stem cell technologies is that we can also use them to generate highly complex 3D organoids, which are small 3-dimensional bundles of cells that can start to capture the complexity of organ systems. And here we can see that we are able to do this in the central nervous system and generate brain organoids that capture the 3D complexity of the brain and mimic a lot of the layering that happens in the brain and the distinct cells and unique cells that happen in the brain. And so not only are these induced pluripotent stem cells allowing us to capture these unique cells in a really elegant model and elegant system, they're also allowing us to capture the 3D structural complexity of these organ systems and provide an incredibly powerful platform, again, to unlock some of the mysteries surrounding the development of these diseases and conditions, but also possible ways to treat them. In addition to the induced pluripotent stem cell technologies, we also have our direct conversion models, which is a new exciting technology that still starts with those same fibroblasts, those same skin cells that we discussed at the very beginning. In this case, instead of going all the way back to the earliest stages of human development and going through the induced pluripotent stem cell process, we're skipping it and kind of moving across the hill. Still using a similar approach where we can introduce a handful of key genes that can induce a fibroblast to change into another cell type. And in this case, we're using genes that can induce that fibroblast to turn into a neuron. And we call this a direct conversion technology. And sometimes we'll refer to the cells we generate from this as induced neurons. Through recent research that's happened here at the Salk Institute and Dr. Rusty Gage's lab, we've learned that when we go through these direct conversion models and generate these induced cell types, that we can capture a lot of the effects of aging. And it just so happens that when we go through the induced pluripotent stem cell modeling process, that technology rejuvenates the fibroblasts that we start with, and we ultimately end up with something that looks kind of young at the end of the day. But when we go through these direct conversion technologies, starting with the fibroblasts and directly converting it into a neuron, we capture a lot of the effects of aging in those cells, which can be incredibly impactful in the context of studying neurodegenerative conditions like Alzheimer's disease. So at the end of the day, we have two very unique and powerful model systems that start from participants in the longitudinal study who donate a small piece of skin through that punch biopsy procedure. We can generate the fibroblasts from those. Those fibroblasts can be used to generate induced pluripotent stem cells, which can be used to generate complex organoids or a myriad of distinct cell types that are involved in Alzheimer's. Alternatively, we can use those same fibroblasts and directly convert them into neurons or potentially other cell types involved in Alzheimer's disease and utilize that technology to really capture age-related effects that are happening with the progression of the AD. At the end of the day, we can use these incredibly novel cell-based modeling approaches as tools to accelerate the discovery of Alzheimer's disease research by unlocking previously unknown disease mechanisms and also providing incredibly elegant platforms to screen new therapeutics and new drugs without doing that in an invasive way in a patient population. It is important to note that none of this research is really possible without participants, and that the Shiley-Marcos Alzheimer's Disease Research Center is actively recruiting new participants into the longitudinal research study that we use for some of the cell-based modeling activities. If you are at all interested in learning more about the longitudinal study and how you could possibly be involved in that, please use the contact information below to reach out to the center for more information. Thank you so much.
We hope you've enjoyed this presentation from our UC San Diego Shiley-Marcos Alzheimer's Disease Research Center Virtual Brain Blast series. Please look for other presentations in this series and share them with your communities. We hope that you'll also consider participating in brain research studies. We always have new opportunities for participation and are actively seeking individuals 65 and older without memory disorders, as well as those with a diagnosis of mild cognitive impairment or Alzheimer's disease and related dementias, such as frontotemporal dementia, Lewy body dementia, and Parkinson's dementia, as well as others. We're also proud to have a bilingual, bicultural team who conduct studies, visits in Spanish. Your participation can make a real difference for future generations. Please click on the description below to complete the very brief survey about the presentation you just viewed. And for those who are interested and who live in San Diego, we'll be happy to contact you. Simply provide your contact information on the secure survey. Thanks for watching, and we'll see you next time.